The overall goal of this procedure is to genetically engineer human pluripotent stem cells using modern genome editing technology.
Though this procedure can provide insight into stem cell biology, it can also be applied to facilitate human disease modeling in a genetically defined setting. Generally, individuals new to this method will need to practice isolating colonies before becoming proficient. Visual demonstration of this method is essential as identification and picking colonies can be difficult.
Begin by culturing human pluripotent stem cells in hPSC media on a 6-well plate containing mitomycin C inactivated mouse embryonic fibroblast or MEF feeder cells grown on gelatin. Each subsequent day after plating, until the hPSCs reach 50% confluency, use a glass pipette and vacuum to remove the entire volume of media. Replace with 3 milliliters of warm hPSC media per well. 1 day before targeting, remove the hPSC media and add fresh, pre-warmed hPSC media supplemented with 10 micromolar Y27632. Also, prepare 1 to 2 6-well plates of drug-resistant MEF feeder cells from DR4 mice. On the day of targeting, prepare the transfection solutions by pipetting 5 micrograms of zinc finger nuclease expression plasmid 1 and 2, talon 1 and 2 expression plasmid, or 15 micrograms of the CRISPR-Cas9 PX330 encoding plasmid into a 1.5 milliliter tube. Add 30 micrograms of the repaired donor plasmid, followed by enough 1x phosphate buffered saline to bring the volume to 300 microliters. Inspect the cells under a